Dhanashree madam.
Yes.
Please.
Good afternoon to one and all, and welcome to this 5 days e-FDP and e-SDP program on evaluation in teaching, guiding values in learning and social understanding through the research. I welcome Dr. Mrunal Patil ma'am, our today's first speaker. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosai, College of Pharmaceutical Education and Research, and all other delegates. Our today's speaker for this session is Dr. Mrunal Patil. Ma'am is Amidex Post-Doctoral Researcher at University of Aix-Marseille University, Marseille, France. As per as the academic is concerned about the ma'am has done her master's and degree from North Maharashtra University in 2010 and 2012. In master's, ma'am is university topper. Then ma'am has done her diploma in patent law from Nalsar University, Hyderabad, and in which ma'am has received 6th rank. Then ma'am received Norton Bhabha Fellowship from University of Nottingham, UK in 2017. Ma'am has completed her doctoral from Institute of Chemical Technology, Mumbai in 2020. And since 2019, ma'am is working as postdoctoral researcher at Aix-Marseille University, Marseille, France. As per as the rewards and achievement concern, ma'am has awarded with a title Unique Designer in 2015. Then ma'am has got first prize at Bio India in 2015, which is an entrepreneurship workshop organized by Biotechnology Enterprises. Then ma'am has also received the second prize in Based India 2014, which is also an entrepreneurship development program, which is organized by ABEL and DBT, Government of India. Ma'am is university topper in her master's in 2012. As per as the grants which is received as per as the grant concern which is received by ma'am has received 2 travel grants, one from CSIR in 2018 and second from ICMR in 2016 to attend various conferences. Then ma'am has also received an PhD fellowship, that is Newton Bhabha short-term PhD placement fellowship in 2017. Ma'am has also received another fellowship, which is for 5 years by DST Inspire in 2013 for her doctoral research. Then ma'am also received junior research fellowship in 2010 for her master's. As per as the research area which is considered, then ma'am has done research on lipid-based nano drug delivery system and antimicrobial drug. Then the research output from this research is ma'am has 1 patent on her credit, which is under examination. Then ma'am also has 3 different papers in international journals with good impact factor and 6 conference abstract on her credit. With this short introduction, I welcome you, ma'am, and also request you to talk for today's session on scientific know-how of antimicrobial research. With this, I request Mrunal Patil ma'am to start her session. Please, ma'am.
Thank you for the warm welcome. So I would talk about scientific know-how of antimicrobial research. As we all know about antimicrobial research, but we do not know how to process or how to progress with doing antimicrobial studies. This whole presentation is basically for those people, postgraduate, graduates, and the researchers who want to pursue their careers in antimicrobial research. So the content of my today's presentation is, first I'll be talking about what is the importance of antimicrobial research. In this we'll talk about drug resistance. then technique to combat drug resistance. Then we'll talk about essentials in antimicrobial testing. This is the core part of my presentation and the most important part. Here I'll be talking about what all things you need for testing your compounds for antimicrobial studies, and as well as what are the techniques involved to test. Then not only a single molecule, but if you want to check molecules in combination, how you should proceed with testing of 2 or more molecules. Then I'll be giving a very short highlights on microscopy, because microscopy is an integral part of antimicrobial research. So here today I'll be only talking about 3 microscopy which is widely used, that is transmission electron microscope, TEM, scanning electron microscope, SEM, and atomic force microscopy. So to begin with, what are antimicrobial agents? It's a very broad term, antimicrobial agents. So basically antimicrobial agent combines 4 different categories. First, antibacterial. The classic example is penicillin, used for staphylococcus and streptococci infection. The next is antifungal. Best example is fluconazole for candida infection. Then antiparasitics. Benzonidazole for Chagas disease is a good example. And antiviral. I am sure till now everybody knows antiviral, since we are going through a very bad pandemic. But a classic example is acyclovir for varicella zoster, chickenpox. Apart from this classification, there are more 2 types of classification, which is a general classification. First is static antimicrobial agent and cidal antimicrobial agent. When I say static antimicrobial agent, that means it is not killing the microorganism. It is just arresting or preventing its growth, that is multiplication. So 10 microorganisms will not multiply into 50 or 60. They will remain 10, but they won't die. So example of this is chloramphenicol and clotrimazole. Then the next category is cidal. In cidal, the molecules actually kill the bacteria or fungus, the microbes that are under test. Example of this is terbinafine and vancomycin. The other category that we can divide antimicrobial agents is narrow spectrum and broad spectrum. In narrow spectrum, the antimicrobial agent can kill only one type of population For example, penicillin G. Penicillin G can only kill gram-positive bacteria. It is ineffective on gram-negative bacteria. Whereas broad spectrum, they can kill a very large group of microorganisms. Tetracycline is one. They can kill gram-positive as well as gram-negative. Amphotericin. Amphotericin is a gold standard. It can even kill a lot of type of fungus, as well as sometimes they're even active on bacteria. So what is importance of antimicrobial research? The key importance that today I'll talk about is drug resistance. This is the biggest problem why we need to continue the field of antimicrobial research. Because whenever we are bringing a molecule in the market, the molecule after some years are no longer effective. So when a therapy, any therapy, antibacterial, antifungal, if it doesn't work on the organism, then that therapy is called as drug resistant. The therapy becomes drug resistant. The organism is no longer affected by that kind of drug. So what is the reason of drug resistance? Well, it's a vicious cycle. There are a lot of population ecology, means a lot of type of different microorganism. It's like a community. You just don't, if I say candida, there is no one candida. It's candida albicans, candida parapsilosis, candida tropicalis, so many species. So the population is ever growing. It's just expanding and expanding. So why this is happening? It's because of the socio-economic growth. We have different kind of behavioral activities. For example, if I take self-medication, if we have cold and flu, despite being pharmacists, we do take antibiotics, right? We take self-medication, but we don't know that eventually these antibiotics will no longer work on us. Even when we are having poultry products, milk, dairy products, these all animals are given antibiotics in their daily food, and this is how it is entering the chain. So we are unknown of this factor, but this is building a tolerance level for the microorganism, and hence the microorganisms are evolving. There is a resistance for those drugs, and those resistant bacteria, fungus, mycobacterium, they all contribute to new population. Hence the population ecology is growing day by day. So drug resistance, when I say drug resistance, there are two types. First is intrinsic drug resistance, means the microorganism is naturally resistant to some drug. Like I told, penicillin G only works on gram positive bacteria. It does not work on gram negative bacteria because there is no target. Penicillin, they work on peptidoglycan, and there is no peptidoglycan or less peptidoglycan in gram negative bacteria. So basically penicillin is ineffective against gram negative. So lack of target. Then there is efflux mechanism. Efflux means where the drug is thrown out of the microorganism through pumps. There are pumps inside microorganism. They throw out the drug. They don't let the drug go inside the microorganism. Then drug inactivation. There are certain enzymes inside the bacteria or the fungus which inactivates the drug. So this kind of mechanism contributes to intrinsic resistance. Now, some are acquired resistant. Acquired resistant takes a little bit of time. That means eventually throughout the year, a microorganism will get familiar with your drug, and it will gain knowledge about the drug and try to modify itself. The modification can be through transformation, transduction, conjugation. It's like genetic modification. The genes will change. They will become more virulent. They will change their genetic combination so that they can counterfeit the drug which is present. So this is acquired resistance. And the major factor, major factor, and I can say last 10 years, this factor is the major reason responsible for resistance, that is biofilm. Now, what is biofilm? Biofilm, if you are leaving a glass inside water, it gets sticky and slimy. Have you ever touched that glass? The sticky and slimy thing is nothing but biofilm. So the microorganism, they're very smart. They attach to a surface and they start building a cover around themselves, just like raincoat. If it's raining, the water doesn't enter your body, right? So for microorganisms, the same. It's a raincoat for all microorganisms. They put a covering on themselves. No matter how much ever drug you put, the drugs really do not penetrate this microorganism. So these biofilms are super resistant. That means they are 100 to 1,000 times more resistant than the actual dose. If you want to if you want to remove biofilm, you have to put 100 to 1,000 times more of the actual dose that you're giving, which can be toxic. So hence this is the current problem of antimicrobial therapy. And if you're looking for a future, I guess this is the best topic to work with. Now, where these biofilms occur, body implants, dental, those plaques that you have on your teeth, they're nothing but biofilms. Then catheters, saline catheter, urinary catheters. These are the places where biofilms are commonly found. Then how bad is the situation of antimicrobial resistance? Trust me, just as a highlight on India, one child dies every 9 minutes. This is the situation of antimicrobial resistance. And hence it is very necessary that the research has to be boosted to overcome this. And what we are losing globally, a failure to address this problem of antibiotic resistance, by 2050, there will be 10 million deaths, and it will cost us more than $150 million annually. So can we fight resistance? Yes, by investing in antimicrobial research. So I have just roughly combined the approaches that we can use to fight resistance. The first is, of course, new drug discovery, but we know it is very time consuming and it is expensive. for a drug to enter and market, it takes more than 14 years because you have to go through patenting system. The patenting system itself takes 12 years, and then marketing. It's an expensive affair. Then biopharmaceuticals is the same thing. Biopharmaceutical products are new products eventually, and they are time consuming and costly. Cationic peptides is relatively a new stream. People are working on peptides, and they are also treated as new molecules. Well, then the other three, that is nanomaterial, drug-drug combination, and adjuvant drug combination, is something that you should look out as a researcher. Nanomaterial, I'm sure people know about silver nanoparticle or copper grids, copper nanoparticles, nanofibers. So these are nanomaterials, and these are finding now new pace. And these materials are used either in combination or in alone to fight drug resistance. Then we talk about drug-drug combination. Drug-drug combination can be a bit tricky if toxicity comes in focus, because two drugs can affect each other. Maybe they are synergistic when you test them, but inside your body, it can alter a lot of mechanism. So drug-drug combination can be tricky, but yes, there are a lot of drug combination which are in phase 3 clinical trials. And I'm sure that maybe after 5 or 6 years, many of them would be in market. The next is adjuvant. Now I have highlighted adjuvant here because I want you to know what is adjuvant. Adjuvant is something which is not drug. Adjuvant means additional. So they are given in combination of drug. They may be active or may not be active, or maybe they actually have very substantial activity, like a little bit low than the actual drug. So, for example, I gave copper and any drug. Silver, we have burn therapy. We for burn therapy, there are some creams and gels in the market which are with silver. So this is adjuvant therapy. If you see the antimicrobial activity of silver, the MIC is quite high, but when you use in combination, they're synergistic. So adjuvant is something which is other than drug. So now people are doing a lot of research in adjuvant and drug combination. Okay, so I start with the main part of this presentation is how do you proceed with antimicrobial susceptibility testing? Antimicrobial testing is also known as AST, antimicrobial susceptibility testing. So first step is know what you're going to test. That is, know your microorganism. So the first question you ask to yourself is whether you are going to work on fungus, a bacteria, or a mycobacterium. The next step you take is check the biosafety level of those microbes that you have chosen. Because we have 4 type of biosafety level, BSL 1, 2, 3, 4. BSL 1 microorganism, you can work on bench between 2 burners. Like we do in bachelor's, we take 2 burners and we work on the platform. So BSL 1 microorganism, these are relatively not that hazardous. That's why you can work on bench. BSL 2, they can be worked on bench, but you have to have good hands on your microbiological testing, and you should wear some protective clothes like apron, gloves to do this. Or you can carry these experiments in biosafety cabinets. Biosafety cabinets look like this. They are closed cabinets with air continuously flowing through it. You have HEPA filters inside. So biosafety level 2 microorganism should be always done preferentially in biosafety cabinets. 3 and 4 are the most dangerous biosafety level. 3, TB, Mycobacterium tuberculosis, falls in class 3. So BSL 3, you need to have specialized clothing, a controlled room where you have 2-door system, and then you have a directional airflow, and they have to be compulsorily done in biosafety cabinets. Even the biosafety cabinets are different. The airflow is usually filtered and it's usually given out and new air is taken, so it's not recirculated. Now, biosafety level 4 is the most dangerous one. all kind of virus, mostly like Ebola, swine flu, bird flu, COVID, they all fall under this category, biosafety level 2. Here you have jacketed suits with air inside. There is always a positive pressure so that the air does not comes to you and you are not infected. And the cabinets are also different. You actually do not touch the sample. You have to put gloves inside. There are gloves which are from outside and which goes inside, and you have to work inside. And double autoclaving is done for all the samples, and there is always filter air circulated inside the cabinets. So you should know what kind of microbe you are dealing with. The next consideration is whether the microorganism you are dealing with is aerobic or non-aerobic. Because aerobic, you can work in cabinets or outside on the bench top. But if your microbe is anaerobic, you have to use these kind of special instruments where you don't have oxygen. So either you have like a chamber where you can put these bags of CO2 inside and place your plates, or you have such cabinets where the oxygen is flushed out and there is CO2 inside, and you can work at the station. So you should know what kind of microbe you're dealing with. The second most important thing is know what is your drug or your molecule. When I say know what is your drug or molecule, that means you should know about the physicochemical property of your drug. Very important. You should, like we do pre-formulation studies, right? The same way for antimicrobial testing, you should know, pre-know the properties of your compound. Check the pH sensitivity of the compounds. Most of the time when you're doing antimicrobial testing, the microbe produce lactic acid. So it changes the pH of the compound, and if the pH of the media, and if your chemicals precipitate in presence of lactic acid, it can be a problem. You can misread the MIC. The next is thermosensitivity. Now, many bacteria grow at different bacteria, fungus, they grow at different temperatures. So you should also check the thermosensitivity of your compounds, especially the compounds are solubilized before testing them. So the solubilized state should not be thermosensitive. The next and the most important, I recommend everybody to test this, is check the solubility of your drug or molecule. This is often that people neglect. They start with antimicrobial testing and later they know that when you put your molecule inside the media, your molecules are not compatible with media and it precipitates. Media has a lot of salts. They have salts, they have proteins, they sometimes have amino acids, they have glucose. So you should always check the compatibility and solubilization of your compound inside the media, as well as you should know what is their general solubility. First, hydrophilic or water-soluble molecules are usually easy to tackle. The problem occurs when you have hydrophobic, that means water-insoluble molecules, or oils, essential oils, lipids, extracts. So this area can be a problem. Now, what can you do when you have hydrophobic molecule or essential oils? Well, you can use solvents. You can use solvents like DMSO. But remember that the final concentration of DMSO inside your test sample should be less than 1% or 1 microliter per ml. In contrast, what if my molecule is not soluble in DMSO? In that case, you can use surfactants, but you should be very careful with testing surfactants. Usually, you can find a lot of literature mentioning 280 at a concentration of 0.01 or 0.1. But in some cases, you can also use non-ionic surfactants like Cremophor-EL, Cremophor-RH40. But make sure that you keep a control for those and use as less as possible. Then an important note, and now this note probably you cannot find in any books or journals. This is my personal experience, that imagine that you are working with DMSO, your compounds are not soluble and you're making a stock solution in DMSO. Now, when you're taking from the stock solution and diluting in your media, your compound may precipitate. Why this thing happens? This may happen because of saturation solubility. So what should I do if my compound precipitate when I suddenly take some amount from DMSO DMSO stock and put it in my media or water? You can gradually decrease the DMSO. Don't suddenly decrease the DMSO. Maybe you prepare a stock solution of 10,000 ppm and then you're directly preparing 100 ppm. So don't do it from directly. If your primary stock solution is 100% pure DMSO, try to reduce it. Make 500 ppm and 50% DMSO, then 30, then 20, then 10. So slowly reduce DMSO so that you don't get sudden precipitation. Then the next thing is extract. I know many pharmacognosy people, they make extract in other than ethanol. There are some, they make extracts in some other solvents like hexane. You can blot it on filter paper disc. If it's evaporable solvent, you can let it evaporate and then put the filter disc for testing. Or there are other techniques which I will tell you as we proceed with our presentation. Next is check experimental protocol. What we do generally is we take a paper, any research paper, and we try to do our experiment according to the research paper. Don't do this, because research paper will give you only short procedure for your experiment. You should always refer standard guidelines. There are 7 standard guidelines, but the most used standard guideline is CLSI and UCAST. Mostly I refer CLSI guidelines because if you can see, most of the papers will give you a reference of CLSI guidelines. And the best part of CLSI guideline is it is validated for microbes which belongs to animal origin. And there are criteria which is given inside the protocol. So I will all give examples. Hereafter, I'll all give examples from CLSI guideline that I follow. So here are 4 CLSI guideline that is used for yeast, filamentous fungi, aerobic bacteria, anaerobic bacteria. There are more. You can browse on internet. You can get several guidelines by CLSI. So these are the most commonly used one, and that's why I have put it in the table. You can refer this. These are available online, and if they are not, you can buy it, purchase it from CLSI website. Now, what do this guideline have? The question is, why cannot I follow research paper? Why do I have to refer guidelines? Well, the guidelines tell you what media should be used, what supplements should be used for a specific microorganism, then how to prepare a stock solution. If it's a known antimicrobial agent, they give you how to prepare the stock solution for known antimicrobial agent. If it's not a known antimicrobial agent, maybe it's your synthesized compound, you can still follow these preparation techniques. Like, for example, this is a snapshot that I have taken from M27A2 guideline, which is for fungus like Candida, yeast. So you can see the first one is for water-soluble antifungal agent. You can see they have given how to prepare the concentration and how to make the final stock solution, how much media to add, how much volume of the concentration to add to get a concentration that is required. And if you can see, they started from 64. Usually we don't start like 100, 50, or maybe 100, 50, 25. We don't start like this because you will end up with decimals. So what we do is we start up with 64 or 128, 32, so that every time you have actual number and you don't have so much of decimals. Okay. And the next is for water insoluble. You can see they have used DMSO here. You can see again they have mentioned dilutions. So you can follow these guidelines. This is for yeast, but if you refer for bacteria, there also they have given tabular form how to dilute compounds. Then again, there are procedures mentioned. Step by step, they have mentioned how to add, how much to add, and how much dilutions to do, how much inoculum to add. When I say inoculum must be a new word to you. Inoculum means microbial cell suspension, the cell suspension that you prepare. So how much to add, how much quantity of cells should be there while testing. These all preparation techniques are included. And the most important is how to interpret your results. And the most important thing, which is not given in the research paper, is quality control standards. There are standards. How do I know that if I have done some experiment, they are correct or not? So you should always keep quality control standards and see there are MIC ranges given for each antifungal agents here. So you can see if you have done your experiment correctly, you will find your MIC in this range. That means you have done your experiment correctly. The next thing is breakpoints. Breakpoints means nothing but MIC. In a general term, microbiological term, we call it as breakpoints. This is not much useful if you're testing your compounds. This is useful for the people in hospital who want to know that, oh, I isolated one microorganism from a patient, but is the patient having drug-resistant microorganism or it's just normal microorganism? In that case, what do doctors do is they isolate the microorganism from the blood or whatever sample it is. Then they culture it on the plate, and then they proceed for antimicrobial testing, and then they refer to this standard. Imagine I test a lot of antifungal agents for maybe I isolate candida from a patient and I test a lot of antifungal agents. If the MIC for fluconazole is less than 8, look here in the table. If the MIC is less than 8, that means it is a susceptible microorganism. But if it's between 16 and 32, that means it's dose dependent. If you increase and decrease the dose, maybe they'll render susceptibility. But if it's more than 64, that means it's a resistant type of microorganism. This interpretive guidelines are also given in the protocols that I mentioned. The next is step 4, know which method to adopt. Often there is a confusion that which method should I go with? There are a lot of methods. Like you have diffusion, Stokes method, microdilution, macrodilution, Agar method, e-test. E-test means epistolometry test. So there are qualitative and quantitative methods. So the most important thing, I won't discuss all the methods here. I will discuss only these 3 methods: diffusion, broad dilution, and Agar dilution, because these are the most repeatable and reliable methods according to CLSI guidelines. So in today's presentation, I'll be discussing these 3 methods. First method is diffusion, Kirby-War's method, as we popularly know. So this is the most easiest method to do. The first thing that you do is grow microbe overnight. Do not skip this step. Skip this step. You should always have fresh microbe. That means culture your microbe overnight and have fresh microbes. They should be always in the exponential state. If they are at dying state, they'll be very weak and you won't get the right results. Then the next thing is you make a cell suspension, what I call as inoculum. So cell suspension you can make by 2 methods. The first method is you can just suspend some colonies inside saline and match it with McFarland turbidity standard. Now, what is McFarland turbidity standard? It's nothing but a test tube which has barium sulfate inside it, and barium sulfate produces some kind of turbidity. You get this McFarland standard in Sigma, you can get it from High Media, you can order them. So you have to match the turbidity of your cell suspension with McFarland standard, which will be roughly equivalent to 150 million cells. And then dilute it as per the protocol says. Some protocol will not have 150 million cells. It will say, oh, just make 50 million cells. So you have to dilute accordingly. So the cell suspension, why I say refer protocol, because for all microorganism, the cell quantity that goes in the test is different. Then you spread this cell suspension. Put few microliters on your plate and then spread it with the help of spreader. Another method that you can use is cell suspension in molten agar. Here we suspended it in saline. Now we are suspending, it's an alternative method. Now we are suspending directly inside the agar. We are not spreading on the agar. We are directly putting the cells inside the agar. Here, the most important point to note is your agar should not be super hot. If it is super hot, the cells will die. It should be hot enough just to touch. It should be warm, basically. And then pour the agar into the plate. What is the difference between two techniques is in the first techniques, the bacteria or the fungus will grow on the surface. And on the second technique, it will be embedded inside your agar. Then what you do is, if you're using known antibiotics, you get discs of known antibiotic agent. You can order it and you can use this kind of dispenser to dispense directly. No problem. If you don't have dispensers, you can use forceps to put this disc. So this technique is called as disc diffusion. Alternatively, you can also use cork boring technique. Cork boring, you have cork borers like this. You have to sterilize the tip of the cork borer, wait for it to cool. Do not directly use hot cork borer on your Petri dish because it may damage your Petri dish as well as it may damage your agar. So do not use direct hot cork borer. Let the cork borer cool after heating it. Then you make holes, and in these holes you can put your antibiotic agent or you can put your test agents. The next test is you can even use this technique with ipsilometer. Ipsilometer, like we have disc here. Instead, now if you want to test 10 ppm, 20 ppm, 30 ppm, 40 ppm, you have to put so many discs, right? Now we have ipsilometer, which have a gradient of your test agent loaded on it, gradient of antimicrobial agent loaded on it. You can see here 32, 24, 16, 12, 8, 4, 6. You can just put the strip onto your agar plates that you have made and then check the reading. The lowest where you see the point where there is an inhibition, starting of the inhibition is your MIC. So how to interpret this data? Sorry, before that, I would like to tell you that this particular technique is very useful for plant extract. Why I say this technique is useful for plant extract is you can directly absorb the plant extract on the disc and allow it to dry and then directly place on the Petri dish. Data interpretation, you can measure in centimeters or millimeters. You use normal scale to measure it or use vernier caliper. And you measure the diameter of the zone. You can see there is a clearing in this image. You measure how much there is clearing and you can report. There are guidelines which tells you how much centimeter clearing should be there for a particular antimicrobial agent. You have guidelines also include this. So you can match it up with the standard. Now, important note is this method is applicable only if your compounds have diffusibility. Now, you may ask me what is diffusibility? Like if I put a powder will not diffuse into agar. A solvent, maybe a solvent or a liquid form will have certain diffusibility. So if you have diffusibility of compounds, you will get a zone. But if there is no diffusibility into the agar, you will not get a zone. So this can be a problem. Sometimes some of the solvent don't diffuse inside the agar. And that's why we recommend that usually the compounds where you can solubilize your agent completely, only such compounds should be used for zone of inhibition assays. And the most important thing here is you should know the inoculum that you're spreading onto the plate. The inoculum that you're spreading should be equivalent for all the plates, your control plate, negative control, positive control, and the test. So this is the most important point. And the most important point is depth of agar. This is relatively neglected by everyone. What we do is we prepare the agar and we hold the Petri dish and we directly pour without even knowing how much we are pouring. So I recommend people that you make agar in different conical flasks And each conical flask should have equal volume of agar and pour the entire content inside the petri dish. Or if you have agar dispensers, then you can pour through the dispensing equipment, you can pour the agar. But each plate should have the same agar, because the depth of agar, your compound is not going to diffuse only horizontally. It will also diffuse vertically. So if the depth is more, you can get differential reading. So every time you repeat the experiment, it should be same amount of agar in the petri dish. And also test the pH. As I earlier said, there may be some precipitation because of pH. The only advantage of this technique is it's very cost efficient and it's very easy to modify. The next and the most important testing method is broth dilution. There are two techniques in this, macro and micro. First, we'll talk about macro. Macro means the volume is more than 1 ml usually. So how do you do it? The first step is prepare test concentration. Now, very, very important point. When you are preparing test concentration, it should be twofold. That means if I require some concentration, I always prepare twofold higher concentration than the desired concentration. So now I prepare primary stock to 2560. Then I dilute it 1:10, I get 256. This I do only to reduce, if I have DMSO in it, I will reduce the concentration of DMSO. So I'm starting with 256. And in other test tubes, this assay is performed in test tubes because the final concentration is going to be 2 ml. That's why you can take glass test tube for this. You put 1 ml of media in each test tube, growth media. And then what you do is your first concentration is 256. That is your stock solution. Now you take 1 ml from your stock solution, you put in first test tube. Now you see it was 256, it reduced to 128. Why? Because there is 1:1 dilution. Already there was 1 ml of media and now you added 1 ml of your stock. So there is 50:50, 1:1 dilution. So 256 became 128. Now you again withdraw 1 ml, put it in the other test tube. Again withdraw 1 ml, put it in another test tube. Every time, and just the last one you discard. So you can see I have a range of concentration, 128, 64, 32, 16, 8, 4, 2. Now when you're prepared the test concentration, this is twofold test concentration that you have prepared. The next, you prepare inoculum. That means cell suspension. Cell suspension you can prepare two ways. Either you match it with McFarland standard or you measure it with optical density reader. You have readers for measuring the turbidity. So, but for this, the second technique, you should know what is the equivalent factor. That means how much optical density corresponds to how much of cell. So that you have to do it. You have to pipette from every optical density, plate it on agar, count the bacteria. If you don't want to fall in this, mostly all microbiological lab already have established the equivalent factor for optical density of all microorganisms. But if you don't want to do this, the best way to do it is McFarland standard. And then you can have this amount of cells inside it, and then you can adjust. So this also you have to prepare twofold. Now remember, I repeat again, the test concentration are also prepared in twofold and the inoculum is also prepared in twofolds. That means double the strength. So next, you add 1 ml of your prepared inoculum to the test tube having your concentration. Now here each test tube was having 1 ml and the concentration was 128, 64, 32. Now after you add 1 ml of your microbe, the concentration changed. Why? Because there was again 1:1 dilution, 64, and that is why we prepare twofold concentration of of test concentration. Now 128 became 64, and the final volume is 2 ml. So the test concentration, your test concentration is 64, 32, 16, 8, 4, 2, 1. Now you incubate this inside an incubator. The next day, you see the results. Oh, I forgot one thing is you include positive control, negative control, and solvent control. Positive control, 1 ml of cell suspension, 1 ml of sterile media. That means there should be no growth in this. Negative control, sorry, there should be growth in this. Negative control means 2 ml of sterile media. That means there should be no growth in it. This is to identify what if your media was already contaminated. Then the next is solvent control, just to make sure that your solvent is not having any activity. The next day you find MIC. You see here I have given a diagram. In 421 there was growth. In 8 there was no growth. So the very first concentration where there is no growth, minimum inhibitory. This is minimum concentration where you have inhibited the microbe. So this is your MIC. I have given here figure how do macro broad dilution looks like. You can see at 8, it is so clear. 4 is turbid. So this is minimum concentration where we did not find any microbial growth. So that is your MIC. But is the MIC, this is just the MIC. That means we don't know if it is static or it is cidal. To know if it is static and cidal, we do this. We plate. What we do is take some amount of your media from test tube 4, 8, 16, and 32 and plate it on agar. You can take 20 microliter and then spread it on the agar and incubate it for another 1 day and then check the growth of colonies. Of course, now here it was already growth, so there are colonies. But you see, this was our MIC, but we still saw 2 colonies. That means it was inhibited, but it wasn't killed. For some of the compound, the MIC and the killing concentration can be same. Okay, but for some of the agent, especially the static agents, you might still see there are some growth. So, but you see 16, there is no growth. So this is your MFC or MBC. That means minimum bactericidal concentration or minimum fungicidal concentration as what you are using. If you're doing bacteria, then it's minimum bactericidal concentration. If you're doing fungi, it's minimum fungicidal concentration. Now in this technique, you should always remember whatever MIC you get is not a pure MIC. It's not an absolute MIC because it's a double dilution. It's a huge gap between, you don't know what is there for 7 ppm, 5 ppm. I don't know. So this is not an absolute MIC. This is like a range of MIC that you get. So how do you determine the range? The MIC that you have got and the next lowest concentration. So that means here it is between 16 to 8. So then you do test in replicates and you can always report ranges for your MIC. The next technique in broad dilution is micro. We saw macro, now it is micro. Micro means only the volume changes there. There the volume was more than 1 ml. Here it is in microliters, like 200 microliter, 100 microliter. Mostly it's 200 microliters. In this, the procedure is same. Just that instead of 1 ml, you're putting 100 microliter of test concentration and 100 microliter of cell. And this experiment is done in 96 well plates. You can see it's similar way as you see in test tubes. You can see here inside the micro well, the clearance and the turbidity, and you mark the MIC the similar way. And the similar way you can test the MFC and MBC also. Now, many of them ask me question, what is MIC 50 and MIC 90? You can see many journals have reported MIC 50 and MIC 90. What do you mean by MIC 50 and MIC 90? MIC 50, as it states, 50% inhibition of growth as compared to control. 90 means 90% growth as compared to control. But tell me, if you see this figure, can you actually tell where is 50% and where is 90%? Absolutely no. With naked eyes, you're never being able to see what is 50% MIC and 90% MIC. So what you do is you check optical density. You read the turbidity of this plate. There are turbidity readers. You can read it on the turbidity reader, or the other technique is use dyes. We have some dyes which can tell you live and dead. So there are like tetrazolium salt, phenoxanthine salts. The most widely used is risazurin dye. Risazurin is blue in color. If the bacteria or if your microorganism is dead, it is blue in color. It will not change its color. But if it's alive, then it will go under a redox reaction and it will change into pink color. So this is how you know the bacteria is alive or dead. And then you can read the entire plate through absorbance spectroscopy and you can get the results. But is it really important to test it? I don't think so. You can even use graphical representation. But I feel there is no utmost need to report MIC 50, MIC 80, or MIC 90. What you should report in your paper is MIC and MFC or MBC, killing concentration and inhibiting concentration. As such, there is no use to mention, there is no logic in mentioning MIC 50 and MIC 90. So, because not all compounds will give you MIC 50 or MIC 90. Sometimes some compounds directly decrease, it directly kills the microorganism. There is no MIC 50 for some of the compounds. So instead of complexing it, rather just give the MIC and MFC. Now, there is one more meaning to MIC 50 and MIC 90, not to be confused. If medically, from a point of view of hospital expert or doctors, if you see MIC 50, MIC 90 means different. I found this answer on a ResearchGate page, which was written by Dr. Olivier Nautel. He said that if you have 100 species of a similar bacteria, say Candida, you have 100 species of Candida or 100 Candida isolates. Isolates means which are taken up from different sources. If you have 100 isolates and you are testing your compounds on those 100 isolate, and if 50% of isolates are inhibited by your compound, then it's MIC 50. If 90% is inhibited, then it's MIC 90. So medically, this can be confusing. So I rather prefer just reporting the MIC and the Cibles concentration in research article. The next is agar dilution. Agar dilution is similar as microbroth dilution. Instead of broth, you are using agar. No difference. You prepare you prepare the concentration, except in here the concentration is not twofold diluted because you're not going to dilute it with anything. Okay, so you prepare the concentration. You put your you let your agar to be cool. Add inoculum. The inoculum is super concentrated. Sorry, you don't add the inoculum. You just make the concentration in the flask. You pour into a plate, preferentially a square plate, which is graded. And in each square, you can put different microorganisms. You can put different inocula in each plate. Like here, you can see at 4 ppm, just to differentiate, I have red microbe, blue microbe, and green microbe. On 4, all the microorganisms grow. When the plate is of 8 ppm, only 2 microorganisms grew. That means it is the MIC for green microbe. For 16, it is MIC for red microbe. So this way you can test as many as possible microorganisms. Imagine in the previous methods that I told you, you were only able to test 1 microorganism at a time. Here you can test at the max in a single plate, you can test more than 30-40 microorganisms, or even sometimes 96 microorganisms. So you can use this spinning tool here. You can just put microorganism, different microorganism here, pin it. That means you dip your pins inside the microorganism and put it on the plate. So each plate will have different concentration, and at a time you can test 96 microorganisms at a time. So this is definitely the fastest technique to study, but it is very laborious because you have to deal with agar preparation. Agar has to be cooled and then poured. And again, many of the compounds, they are not stable in agar. So you have to even check for stability. This is the most recommended method for fast EDS organism. Fast EDS means those which have to be grown in blood cultures or with certain amino acids. So This is the most utilized technique, and it's the most fastest technique among all the techniques. So checkpoints, always make sure that you check the pH of your media. You should include QC and reference standard in your test studies. Solvent control, do not miss it if you're using DMSO or surfactant. Then you should check for contamination. Do check for contamination. The next thing is repeat all the experiment 3 times, because there is possibility. I told you MIC is always in ranges. So you might get different MIC, and that's why you have to report it in range. Now, how to deal with combination? I'm sure people come up with, okay, this was testing just for one molecule. When you have combination of molecule, what do? First, try to understand what is combination. There are 3 types of combination. The combination can be synergistic, the combination can be antagonistic, or the combination can be additive. Pharmaceutical point of view, we look for synergistic combination. That means 1 + 1 is 11, more than you expect. Then it's of use. Just additive combination is not of that use. So you always look out for synergistic combination. Antagonistic, of course, you don't want to give 2 drugs which are absolutely incompatible with itself and they cancel each other's activity. So you should always check for these 3 interaction. How do you check this interaction? We check it by a method called as checkerboard. The first method I'll be explaining is checkerboard. Checkerboard is the meaning of checkerboard means, have you seen chessboard? Chessboard, you have checks inside it, the similar way. And that's why its name came from there, checkerboard. It's also called as fractional inhibitory concentration index, FICI, that we say. Now, here we have 2 drugs, drug or agent, drug A and drug B. Okay, drug A I have given by green and drug B I have given by blue. Now, look out here, the most important point is you have to make concentration which is 4-fold higher, 4 times. In macro broth, it was 2 times. Here it is 4 times. And the cell suspension is 2 times. Cell suspension you don't have to change. Cell suspension remains as it is. But the concentration of your compounds should be 4 times. I have mentioned the concentration of compound A and compound B here. Now, you take 96 well plates. Okay. Now, start horizontally. Horizontally, I have drug A. You put 50 microliter of drug A and 50 microliter of drug B in each. How do you do it? You take from one ependoff, you put horizontally A and vertically B. Matlab, horizontally A will be 64 here, 62 all the way here, 16 all the way here. And for B, it will be 128 here, 64 here, 32 here. There will be one well where you don't have A, where you only have B, but you don't have A. That will be 0. 0, that means you will not put anything. No, you will put plain media. You will put media inside it, but you will not put the A. Okay, this is without the A, and here it is without the, it is without the B. Now, you added 50 microliter of A, 50 microliter of B. That means you did 1:1 dilution. Check here. It was 128 ppm for compound A. Here it became 64. Why? Because you also added B to it. So the final concentration for A became 64. For B, it was 256. Now it is 128. Okay, because you're 1:1 diluting it. 50 of A is going inside one well and 50 of B is going inside one well. The next is, okay, 50 plus 50 is 100 microliter. Now in each well, you have 100 microliter of your test concentration. Now you add 100 microliter of your cell suspension as you prepared. I told you how to prepare microbial cell suspension. You take 100 microliter of your microbial cell suspension and add in each well. Now 100 plus 100, 100 of cells and 100 of this test what you did. Now check here the concentration, 32. It was 64, it changed to 32. It was 128 here, it changed to 64. Now you check this 128 and 32. It is fourfold than the actual concentration used, right? That's why you prepare fourfold than the concentration that you have to check. So if you want to test 32 ppm, you have to make a stock solution of 128. Okay, that's why we prepare fourfold concentration in synergy assay, a checkerboard assay. Now incubate this plate. After you put the cells, go for incubation. The next day you find this result. Now this, what I have marked is your growth. This is an example that I'm giving you. Maybe this is your growth. In positive, you have growth. Negative, it's only media. That means it's clear. It should not have growth. And this is your checkerboard. You know, it's like checks pattern. Now, what do you check in this plate? First, you will check what is the MIC of your compound alone for A and for B. Then you check MIC for compound in combination. How do you, first I'll show you how do you check MIC of compound alone. This is Okay, so I told you that one row and one column we keep no other compound. That means here there is no A and here there is no B, but there is A here and there is B here. So if I want to check MIC of the compound alone, that means here for A it is 8. See, it is clear here and there is no B. So the MIC is 8. Similarly, for B, the MIC is 16 because here there is no A and still there is clearance. That means this is its MIC. Now I know for my checkerboard, the MIC of A is 8 and MIC of B is 16. So don't look beyond the MIC. Look within the MIC, less than the MIC, less than 8 and less than 16. That means this region. You are supposed to look in this region. Now where to exactly look? Look at the border points. These are your border points. Now, for example, I take this point in my border point. There is clearance. Now what is the MIC in this? 0.5 for A. And for this, it is 4 for B. So I use this formula, FICI, and I calculate 0.5 for A and the MIC was 8. 0.5 divided by 8. For B, it was 4 divided by 16. 4 divided by 16. And then I plus it, I get value of 0.3125. Now this is your index, FIC index. Now the value you should match it with this table. If the value is less than 0.5, that means there is synergistic interaction. If they lie between 0.5 and 4, that means it is just additive reaction. And if it's more than 4, that means there is antagonism. So this is how you know whether you have synergism or you don't have synergism. The other technique is to plot isobolograms. Isobolograms, you first put MIC 50. MIC 50 in this case would be 8 is your MIC, MIC 50 is 4. That means half the MIC. You just put here the MIC 50 of both the drugs A and B and you draw a line. This is graphically done. You draw a line and you plot all the points which are here. Okay, the border points, and check where they lie. If they lie below the additivity line, then it's synergistic. If they lie above the additivity line, then there is antagonism. There is one more technique to do it. What if I'm not doing micro broad dilution or macro broad dilution? Then I can do Agar diffusion. In even Agar diffusion, you can see synergism, antagonism, additivity, and autonomy. You can see synergism. It's basically two molecules have combined to make one single circle. That is how you predict synergism. Antagonism, you can see the zone are moving apart from each other. And for additivity, there is merging, but not complete merging. It's not a single circle. It's somewhat merging, just touching each other. And there is autonomy, that means no difference. Absolutely. They're not even running away from each other. They're just no difference. And the most important technique, there is the third technique to find interaction, is time kill. This is widely used in industry and is the most reliable technique to do it. Now what I do is put half MIC in broth. Okay, half, because if you put full MIC, it will kill. So use less concentration than the MIC for A or B. And in different conical flasks, you can mix A and B, half both. And then after every time interval, you take out some aliquot from each flask and put it on the plate, spread it on the plate. So usually you get a graph like this, and you count the colonies the next day. Okay, and if you are counting the colonies, you can put it in a log table and plot it versus time. You find graph like this. Now see A, it was half MIC. That means I'm not killing it. That's why there was a growth. Okay. B also, I'm not killing it. It's still alive. Here is my B. And then I had combination of A and B. If it's synergistic, there will be a tremendous decrease in the cell concentration that you observed on A and B. But how do you find if it is synergistic or not? Well, if there is synergism, the decrease in the cell count is more than 2 log CFU. Okay, if the decrease is more than 3 CFU, that means it is a Seidel effect. So this technique will also give you synergism. It will also give you Seidel effect. Okay, and this is most utilized in industry, or it is used as a confirmatory test to checkerboard. And how do you compare this? You compare your combination with the most active compound that you're using. You can see from A and B, my most active compound was B. So I compare this with this. I compare A plus B with B. And if the difference between A plus B and B is more than 2, it's synergism. If it's more than 3, it's Seidel. Now moving towards microscopy. Microscopy, you have light microscope and electron microscope. I'll not explain in detail. You can just check the difference. But the most important difference is electron microscope uses electrons as an illuminating source, and light microscope uses light. So I'll just come to how they look. You know, if you're observing under light microscope, this is nanofibers. See, it's very blurred and it's not clear. Where if you see in SEM, it's very clear because the magnification is more, the resolution is more. So I'll start with first TEM, transmission electron microscope. It has an electron source, which is often a tungsten lamp. The electrons are emitted from it and they are channelized and focused by electromagnetic lenses. That means only a definite energy, a known energy or definite energy containing electrons will be channelized into one channel, and they'll be focused on a sample holder where you have your sample kept. In TEM, the sample holder is usually a copper grid or gold grid. You can put your sample here on the grid, and then you can place it in the sample holder chamber. This is the sample holder chamber. It is very small sample holder. And then the imaging is done by electromagnetic lenses, and there is a screen where you can see the electron directly. If they bombard on the screen, it glows. And with the help of this, you can see the image that has been obtained by electromagnetic bombarding. Now the reason why we call it as transmission, it's because the electron passed through the sample. That's why we call it as transmission electron microscope. Now TEM needs sample to be fixed. You cannot just place your sample and then let it see. No, it cannot happen. There has to be a contrast to see your sample. Electrons cannot directly visualize your samples. So you have to stain them. You have to fix them. So for biological sample, I will be only speaking about biological sample here. First, you fix it with aldehydes. Aldehydes, formaldehyde is the best. You fix them. nucleus in your cells can be fixed with aldehyde. It's so that it's more prominently visible. Then you secondary fix with osmium tetroxide. Osmium tetroxide is also fixing agent. It's very poisonous. It usually fixes the lipids. Any part of your cell which contains lipids, osmium tetroxide will fix it. Then you have tertiary fixation like uranyl acetate and tannic acid, just to give a contrast. Then you dehydrate the sample. TEM, you don't need water to be present in the sample. That's why you have to dehydrate the sample. You dehydrate by series of ethanol and acetone. Then you embed inside resin like paraffin, or there are some acrylic resin. And after embedding it, you section. You have microtomes. You section very thin, like 17 nanometers section are made for TEM, so that you have better penetration of electrons. And then again, you post-stain it to get contrast effect. And then you image. Now one thing to remember here, in TEM, it is the contrast which gives you the result. So staining is the most important part of TEM. There is two types of staining, positive staining and negative staining. In positive staining, you actually stain your subject. That means whatever you have to observe, you stain it. You don't stain the background. Okay, so the visualization will be like this. The background will be white or it will be light. In negative staining, you stain the background and you want your content to be bright. So here you can see the background is black, but the content inside, you can see there is a distinct circle, white circle. It is because it shows the circumference of your agent. It's the best for globules and nanoparticles, because you see, if you want to see if it's circular, this is the best thing to do. Example of staining agent is ammonium molybdate, uranyl acetate, phosphotensinic acid, osmium tetroxide acid. Now, how do a TEM image look like? You can see here the TEM image. You can see the nucleus. You can see cell wall. You can see mitochondria. A TEM will give you inside structure. Because you're sectioning the biological sample, you get a view of internal structure. This is not possible in other microscopy. This is possible in TEM. And that's why TEM is important if you want to see how does the chromosome look like, how does the cell wall look like, and how do we utilize it in research. You see here I have given Candida example that I have adopted from a journal. This is Candida cells, and you can see they have magnified the cell wall. Can you see the changes in the cell wall? Here the cell wall is thin, here it is more. Here there are some globules which is formed within the cell wall. So if you want to see internal structure, TEM is the best option. The second is SEM. SEM, scanning electron microscope. It is a surface microscope. That means it gives you information of surface. So here the readings are usually, it is also, the source is also electron beams, again tungsten filament. But here the source is obtained from the secondary scattering, the secondary electron scattering and the back scattering and the X-rays which is generated. So it gives you the idea of the surface of the subject which is being tested. It is slightly, the mechanism is slightly different than TEM. In TEM, you just have the electrons which has been captured, the past electron, which is the electron which has passed from your subject. Here it is the scattered electron which is captured, the X-ray and the secondary electron which are obtained from the sample. Now, the advantage of SEM over TEM is you don't need fixation. You may or may not. You cannot, of course, use wet sample. You have to dry the sample, but you do not need any special treatment. You don't need contrasting agents or anything. You can use, but you don't need it. And the best thing is the samples are very easy to place. They don't, you don't need fixation, or you can do fixation, but it's not required. A raised surface will appear brighter, and a surface which is in deep will resemble darker. So this is how scattering works. You can see the same image. You see there is no internal observer. I cannot tell you here where is nucleus, where is cell membrane, because it's a surface phenomena. This is again Candida cells. See how they look. And this is basically a herbal extract which is treated. These cells are treated. You can see there is deformation on the cells. You can see deformation on surface if you want to do. If you just want to see deformation, you can go for SEM rather than going for TEM. TEM is very time consuming and it is expensive. You can rather prefer for SEM. Now, the most important is atomic force microscopy. I'm sure for many of the people this is new. Atomic force microscopy is not an electron microscopy. It is probe microscopy. It does not work on the concept of electron. There is a probe, or you can say cantilever. Okay, this is very minute and it runs through your surface. If this is your surface, it is running through your surface and generating a memory in the photodiode receptor, where you can translate it and then see it in your PC. So advantage is you get a 3D image. In SEM, TEM, you're not getting a 3D image. Here you actually get 3D image. There it's only a 2D image. So this is the best microscopy if you want 3-dimensional view. Then, and the best advantage is this can even work in liquid. If you have any samples in liquid media and you cannot dry the samples, then yes, you can do AFM even in liquid. The disadvantage is it's a very slow process. It takes a lot of time to adjust the area and to scan the area, because the probe has to move over the area and the probe moves very slowly. So it is a very time consuming process and it is affected by non-linearity. And there is a possibility that there may be artifacts. That means the probe can damage your sample. And then normally it cannot measure steep walls and hangings. You can see the AFM probe, if you have a circular obstacle, this much of area will be measured by the probe, but it is actually not the area. It's a void, but it's still measured because it's an artifact. So this can be a disadvantage, but now there are specialized probe and different probes made for AFM, which have overcome this deformity. Now there are modes of AFM: contact mode, non-contact mode, tapping mode. Contact mode, there is a direct connection of the probe with the sample, but here your sample can get damaged because it's in direct contact. Non-contact, there is low resolution, but your probe do not touch the surface. Then there is tapping mode, where there are intermediate contact with your compound or your sample. And the AFM has 3 major abilities. It can measure, it is not only utilized for taking micrographs, not only for imaging, it can be used for other things also. You can measure force. That means you can measure elasticity, flexibility of a molecule. You can do it by force measurement. Then you can, manipulation is one of the technique. Manipulation means if you want to see drug binding assay or binding antigen antibody binding, it can even do that. So biological application, I have mentioned here all the biological application. As I told, it can measure binding forces, ligand binding, ligand receptor binding, antigen antibody. For that you need modified probes. Maybe your receptor ligand is attached to the probe, and then you are checking its binding, and the cantilever deflects upside and it calculates the energy. So energy is always calculated in the form of electrical properties here, and hence you can even measure that thing. So it's a multi-use microscopy. Here I have given an example from a journal which have measured the elasticity of a protein. You know, the protein was placed on glass cover slip, and then the cantilever just elongated it to see its height. So you can see its height, you can see its force, and you can measure how the proteins have unfolded. Similarly, you can even see binding. You can get a data for protein complexes and then interpret how was the binding, how much energy was required for binding. So this is a very unique technique and the latest technique that can be used in biological samples. This is an image from my paper where I did AFM for ultrastructure. You see, this is also Candida cells. You see, there is a graph which has been generated by the machine. Also, I can get a 3D view of my compound of my cells. See the last two where I have treated with combination. You can see there is a depression. I measured this depression. How big is the depression with my combination? So you can actually see the first one is control. It is so plain and so circular. But then when I treat it, it is an abrupt or irregular surface. So you can use this technology to get topographic information or binding information. So with this, I end my presentation. This is my university. We have many campuses in the city. And this is our city. It's Marseille. I know it's written as Marseille, but it is called as Marseille. It's the second largest city in France. And if you ask me where it is to the south of France, very far from Paris. So any questions?
Thank you, Dr. Mrunal. It was very nice and informative session.
Thank you, sir.
Yes. So, is there any question from audience? Mrunal, you explained in very simple manner. I think those who attended this session might got 100% because it's a very basic topic, in fact. And there is one question from moderator itself. I think
Yes,
someone might have asked question. Which method is best suited for new synthetic antimicrobial compound for primary screening?
Okay, if you have a big library, I would suggest you Agar dilution method is the best if you have very big library. But if you have a couple of agents, synthetic agent like 2050 or maybe 2050, then you can go for micro-broad dilution. Here, the most important factor that you have to consider is solubility of your compound. Mostly when the molecules are synthesized, they do not really have good solubility. But I guess most of them work good with DMSO. So I work on synthetic molecules. I prefer micro-broad dilution. Because I have 20 to 25 molecules. So for that, micro-broad dilution is the best. But if you have more, then go for Agar dilution.
Is there any more question from audience? No question. Thank you, Dr. Mrunal. On behalf of Vocal Education Society's Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I am thankful to Dr. Mrunal.
Thank you,
it
sir.
was very good session, in fact. So I am thankful to Dr. Mrunal. I am thankful to Mrs. Dhanashri Mari ma'am for moderation of this session. So thank you. All delegates are requested to tune. The second session will start immediately in next 2 minutes. Thank you very much. Thank you, Dr. Mrunal. Thank you.
Sir. Thank you, sir.